Hi students, today we will see nucleotides. Nucleotides are the monomeric units of nucleic acids for both DNA, deoxyribonucleic acid, as well as for RNA, ribonucleic acids. Okay, this nucleotide consists of a nitrogen base, and a pentose sugar and a phosphate. This is the basic structure of a nucleotide. These nucleotides joins together to form polynucleotides and thereby forming DNA as well as RNA. Okay, now we will see the nitrogen bases. There are 5 different types of nitrogen bases. Okay, first one is NH2. Yes, 1, 2, 3, 4, 5, 6, 7, 8 and 9. This is a purine based nitrogen base. A purine based nitrogen base. This is adenine or 6-amino purine. Then second one, again another purine nitrogen base. This is guanine. That is 2-amino 6-oxypurine. These two nitrogen bases are based on purine nucleus. Okay. Then there is pyrimidine nucleus. Pyrimidine type nitrogen bases. We know the structure of pyrimidine. This is pyrimidine and and nitrogen. 1, 2, 3, 4, 5, 6, 4-amino 2-oxypyrimidine or cytosine. You can call it as cytosine. Then another pyrimidine nucleotide. This is uracil, 2,4-dioxypyrimidine. Then two 4-dioxy 5-methyl pyrimidine. This is also a pyrimidine nucleus. So there are 2 purine based nitrogen bases and 3 pyrimidine based nitrogen bases, cytosine, uracil and this is thymine. Okay, 3 pyrimidine type nucleases. Then in DNA, adenine, guanine, cytosine and thymine. These 4 are the nitrogen bases found in DNA. In RNA, adenine, guanine, cytosine and uracil. These 4 nitrogen bases are seen in RNA. Then coming to the pentose sugar, in DNA it is deoxy ribose. In RNA it is ribose sugar. That is the difference. In DNA, adenine, guanine, cytosine and thymine. These four are the nitrogen bases seen in DNA. Then deoxyribose is the pentose sugar present in DNA. In RNA it is adenine, guanine, cytosine and uracil. And the pentose sugar is ribose. Now we will see an example for a nucleotide. So first I will say there is adenine. See, this is adenine, and a hydrogen is replaced at the 9th position, the nitrogen at the 9th position, and a pentose sugar attaches to the 9th nitrogen. Okay. So this is the structure of a nitrogen base with a pentose sugar. A nitrogen base plus a sugar molecule, a pentose sugar molecule, that is known as nucleoside. And if a phosphate group joins to the nucleoside, it becomes nucleotide. So this is a nucleoside, and this nucleoside can be called as adenosine. Adenine, a sugar molecule joined, it became a nucleoside adenosine. Okay. Then at the 5th position, if a phosphate group joins, it becomes adenosine monophosphate. Or AMP, or we can call it as adenylate. Okay. So this is the structure of a adenine based nucleotide present in RNA. Why? Because it is a ribose sugar. So in the second position, if oxygen, so again I will draw N NH2, adenine, 9th position, sugar molecule joins, CH2, phosphate group attaches. In the second position, here instead of this OH, oxygen is eliminated to form deoxyribose. So deoxyadenosine monophosphate. Deoxyadenosine 5-monophosphate, or deoxyadenylate, or we can call it as deoxyadenylate. This is the case of adenine. In guanine also, this 9th hydrogen at the 9th nitrogen is replaced by a sugar molecule. If it is by ribose, it is guanosine. It becomes guanosine, and a phosphate group attaches, and it becomes guanosine monophosphate. Then similarly, if deoxyribose attaches, then it becomes deoxyguanosine monophosphate. Okay. Then in the case of pyrimidine nucleotides, the hydrogen at the first position, hydrogen at the nitrogen on the first position is replaced by a pentose sugar. So this can be called as Cytidine is the nucleoside, and if a phosphate group attaches, then it becomes cytidine monophosphate. Cytidine monophosphate, and cytidine monophosphate or cytidylate. Okay. Similarly, with uracil, a phosphate group attaches, it becomes uridine. Then a phosphate, if a sugar molecule attaches, it becomes uridine, and a phosphate group attaches, it becomes uridine monophosphate, UMP. Then similarly, for thymidine monophosphate. Okay. Here thymidine is seen only in DNA, therefore we can call it as deoxythymidine monophosphate. Thymidine is seen only in DNA. Only deoxyribose attaches to thymine, and then it becomes deoxythymidine monophosphate. Okay. Here, if this oxygen is eliminated, then it becomes deoxycytidine monophosphate. Okay. This is about the structure of nucleotides. And then coming to the functions of nucleotides, of course it is a part of genetic material, that is DNA and RNA. It's a part of genetic material, and then it acts as a coenzyme. These nucleotides are a part of coenzymes. For example, nicotinamide adenine dinucleotide NAD, then nicotinamide adenine diphosphate NADP, dinucleotide phosphate, then FAD, flavin adenine dinucleotide, then flavin mononucleotide FMN, coenzyme A. In this, these nucleotides are involved. Then it acts as a second messenger. For example, cyclic AMP and cyclic GMP. They act as second messengers for hormones. Then again, they act as metabolic intermediates. For example, UDP glucose. We studied UDP glucose in glycogen metabolism. It's the donor of glucose for forming glycogen, UDP glucose. Okay, it acts as a metabolic intermediate. Then it again acts as a methyl donor for many reactions inside the body. Methyl donor, for example, S-adenosine monophosphate SAM. Act as a methyl donor. Then it acts as a high energy compound. It acts as high energy compounds, for example, ATP, adenosine triphosphate. Okay. Then again it acts a part in glycoprotein synthesis. For example, UDP glucose, UDP galactose, etc. So it acts as a part of genetic material. Then it's a part of coenzyme, example NAD, NADP, FAD, FMN, coenzyme A, etc. Then for in second messenger, cyclic AMP and cyclic GMP acts as second messenger for many hormones. Then intermediates, it acts as metabolic intermediates. For example, in glycogen metabolism, UDP glucose acts as a glucose donor. Then for many reactions, it is a methyl donor. For example, S-adenosine methionine. Then high energy compounds, that is ATP, in its take part in glycoprotein synthesis, for example UDP glucose as well as UDP galactose. Okay, thank you.